Hi, Dr. Biology here. This video is from my vault of older videos. This is actually one of the first papers I went through, which is A-level biology, an old spec paper, but some of it is still pretty much valid in the new spec, and it's related to populations and environment. So that'll be things like respiration, photosynthesis, inheritance, aspects of inheritance, energy transfers, ecology, ecosystems, so all those kinds of things. So it's another resource possibly you could use in your preparation for your A-level. So I hope you find it useful. Again, apologies about the sound quality, but it should be self-explanatory. It's quite a good way of learning. What you could do is pause after each question, think about the answer, even write down an answer, and then I'll go through the answers with you. I hope you like it. Hello Year 13, and welcome to this YouTube clip to talk about your Unit 4 Population and Environment mock examination. Please make sure you use a copy of the mark scheme and use that with your question paper whilst we go through the answers. Okay, so looking at the first question is asking about demographic transition related to birth rates, death rates and the size of population. So the first question is definitely what I would call a learn and churn answer. So it's asking you to describe the changes that occur in the birth and death rates during demographic transition. Over time, what will occur is that both the birth rate and the death rate will fall. So you need to mention birth rate and death rate falling. Okay. But then what happens after that? You can't just say it just both falls. You need to then say that it levels out, or it remains stable. Okay, it stabilizes or fluctuates. For the second question, it wants you to describe the changes that occur in the size of the population during demographic transition. And basically, you need to say that the population increases and then again levels out. And I think many of you were getting the kind of half the question correct, but not finishing off. So population is going to increase, but it does level out. Next question is showing a survival curve for males and females, and it's a maths related question, and all they want you to do is calculate the percentage difference in the average life expectancy for females compared with males, and it does say to show you're working. Okay, so first of all you need to look at what is the 50% survivor rate. So at this point it's at 50,000. Okay, so if we draw a line better than I'm doing at the moment, and then you can see where it transects, and so you can see that the male is going to be there and the female is going to be approximately there. The two figures you're then looking for are basically either 79 or 80 and 84. So that gives us a difference of 5 or 4, and all you then have to do is to divide the 5 with 84, so 5 divided by 84, and in that case equals 5.9%. But there are lots of different answers if you had 4 and 79, for example. Notice it's to 1 decimal place. Okay, if you got 5.95, then you show that second decimal place. Question 1b2, it says suggest one reason. Well, there are many different reasons for this. The main thing is if you're going to give a reason, you've got to name it. So a named disease, for example, or a named risk factor. So things like heart disease, prostate cancer, obesity, stress, exercise, driving, war, smoking, alcohol. Notice in the comments in the mark scheme that you can't get the mark for a neutral answer. So you can't just say whether someone has a better lifestyle or more fitness, or related to they drink more, or what is it about drinking more? It's related to alcohol. So be very careful with that style of question to be specific in your answer. Question 2 is about the nitrogen cycle. They give you a nice simple diagram here, and then they want you to say which process is related to nitrification, and if you've done your revision you will all know that it is R, and that is a simple answer. I think 2b is a bit trickier because they obviously have talked about microorganisms add ammonium ions to soil, and they want you to talk about another process. So what you can talk about is the fact that proteins or amino acids are broken down by saprobiotic microorganisms or saprobionts. In the examiner report they talk about the fact that people knew it was a saprobiotic microorganism, but many missed the second question related to the fact that referring to the breakdown of a named nitrogen containing compound such as protein, simply referring to dead or organic matter or feces, and some limited their answers by talking about nitrogen fixation or denitrification. 2c is quite a nice question. It talks about ploughing and how it aerates the soil. So how would it affect the fertility of the soil? Well, very simply it would increase the fertility of the soil, basically because you're getting more nitrate formed, and therefore there's no denitrification, so there's fewer denitrifying bacteria. Okay, or you could say there's more nitrifying bacteria. It's because your process R is being increased. Okay, so as it says, as you can see the answer there. Okay, right. Okay, 2D. 2 questions there, 2 marks, sorry, related to farming practice. Suggests 2 ways in which crop rotation may lead to a higher crop yield. 2D is looking at crop rotation, how it can lead to higher crop yields. Quite a few answers you can give here, but basically you're growing crops that have plants that have high nitrogen fixing bacteria. Also you've got different minerals and salts and nutrients from the different crops you use, and also you're not going to have pests, residual pests remaining in the soil from year to year because you're using different crops. You could also grow legumes. Remember, legumes contain nitrifying bacteria, okay, so they add nutrients to the soil. And that's something that many students didn't really get in terms of the fact that nitrifying bacteria are in the root nodules of legumes. You could accept, as it would have accepted as well, that there was a fallow year. In other words, you leave it and you don't grow any crops, or using different amounts of ions or nutrients. Question 3 is looking at gross productivity and biomass, and they give you some information. And then 3AI, it suggests appropriate units for gross productivity. I have to say there were quite a lot of answers that were wrong, basically, either missing units of mass or energy, or just energy, just mass, and just not very well done. You need to really revise this section. What you should have done was you should have got basically the following, if I can get the correct thing up. So it's kilojoules per meter squared per year. Okay, alright, so it's got to include energy, area and time. 3A2, explain the decrease in gross productivity as the woodland matures. Again, some of you were kind of getting the wrong end of the stick here. Really, all they're looking for is you to talk about how it reduces photosynthesis. So why does photosynthesis reduce? Well, basically the woodland is mature, therefore there is less light or there is more shading. Quite a lot of students went on to talk about increased respiratory losses, for example, okay, and that's confusing net productivity with gross productivity, so just be very careful with that. 3B is quite a simple answers. Basically it's using your knowledge, so again it's learn and churn, looking at succession. So they want you to talk about pioneer species, firstly, and then how over time abiotic conditions change. You get more habitats or niches, and then you get an increase in number and species diversity of plants and animals. Most students did very well on that question. Now 3C, it talks about using your knowledge of net productivity to explain why biomass shows little increase. So I would probably start off with saying, well, what is net productivity? So this is gross productivity minus the respiratory losses. So the decrease in gross productivity can be related to the fact that there's an increase in respiration possibly, and less photosynthesis. Final question suggests one reason for conserving woodlands. I don't think you need to know very much about A-level biology for this. In terms of conserving, protecting habitats, reducing global warming as a source of medicines, etc. There's lots of answers for that. You could also accept tourism, looking at recreational activity as well. Okay, question 4 is about codominance and also about sex alleles as well. Nice question because it looks at inheritance and it also talks about Hardy-Weinberg. So the first question, explain what is meant by codominant alleles. So the response is that both alleles are expressed in the phenotype. I did notice again when marking that many students were writing lots and lots of information when all it needs is a short, sharp, kind of detailed answer. 4B looks at looking at the stem of the question. It talks about male cats with a tortoiseshell phenotype do not usually occur. Basically, a male cat will only possess one allele. The Y chromosome doesn't carry that allele, and therefore the male cat cannot be heterozygous. Question 4C is looking at doing a cross between a tortoiseshell female with a black male, doing a genetic diagram. This was very well answered by the vast majority of students, understanding what the genotype was, using the Punnett square diagram to show that. One of the key things then, the phenotype, some of you forgot to put the sex of the cat, and therefore your ratio of phenotypes came out as 1:1, which is incorrect. Okay, so you need to make sure that you put the sex of the cats, and then you would have got the ratio of basically 1:1:1:1. Okay, so I think most of you, as I said, got that correct. Well done. Okay, so you can see from the Punnett square, that gives the genotype. The phenotype, remember, is a tortoiseshell female, black female, ginger male, black male. This question was asking you to work out, using Hardy-Weinberg equation, the frequency of recessive alleles for the gene. And it does tell you that 19% of cats had extra toes, they had polydactyly. So basically, if you think about it, the cats, you had heterozygous dominant or homozygous dominant, and that equaled 19%. So in terms of our equation, that includes p squared, and that's pq plus q squared. So we need to find q squared. So we know p plus q equals 1, so therefore we can say 0.19 plus q squared will equal 1. So therefore it must be 0.81, so that's q squared. Okay. And if you work out q, which is what we're trying to find out, the recessive allele, so q equals the square root of 0.81, and that equals 0.9. I think a key thing is to remember to always convert the percentage into a ratio, okay? Alright, so 19% is going to be 0.19. Also, I would write out the Hardy-Weinberg equation. That would help you work out what you're trying to do. I'll be honest, that wasn't the easiest of Hardy-Weinberg, but hopefully you've learnt from looking at my example. Okay, 4D2, again I think some students found this quite difficult, but if you read the question carefully it makes sense. So looking at, it says describe how this would affect the frequency of the homozygous genotypes. Well, we know in terms of homozygous genotypes there's two. There's either homozygous dominant or homozygous recessive. Therefore we know polydactyly is caused by a dominant gene, therefore we know that homozygous dominant increases and homozygous recessive decreases. And it's important you talk about both genotypes. Notice in the question it does say genotypes, okay? So read the question really carefully. Question 5, going on to looking at photosynthesis, they give you a very interesting diagram that you've probably never seen before, and they ask you, 5A, what measurements should the student have taken to determine the rate of photosynthesis? So all the answers to question 5A is looking for is just the fact that oxygen is produced, okay? It's not being taken up, it's being produced by the photosynthesizing algal cells. Question 5B is quite nice in terms of looking at two factors which should be kept constant during the investigation, so kind of how science works. You could have had intensity of light, or the amount or the mass or the number of algae or photosynthesizing cells, amount of carbon dioxide, and also a set amount of time. Could have also had the distance from the light. 5C is checking on your understanding that basically as pH increases, more carbon dioxide is removed for photosynthesis. Some students found that quite difficult in places, okay. Weaker responses often confused carbon dioxide with oxygen and respiration with photosynthesis. Question 5D, it gives you a graph showing the rate of photosynthesis and the wavelength of light, and they're talking about suggesting and explaining why the rate of photosynthesis was low between 525 nanometers and 575. To gain full marks in this question, you need to link the fact that there's less absorption in terms of the plant, less absorption due to a decrease in the light-dependent reaction. Or you could suggest that the wavelengths reflected represented the green light at this wavelength of light. Okay, question 6 is very much related to respiration. 6A, a nice opening question, checking again your understanding of your knowledge of acetyl coenzyme A and how it's formed in the link reaction. So to get the 2 marks, they're really looking for you understanding that pyruvate, there's oxidation occurring and hydrogen is removed from pyruvate and carbon dioxide is released, and the addition of coenzyme A. 6B, they give you quite a bit of information in the stem of the question, and they're talking about the fact that the reaction between acetyl coenzyme A and oxyl acetate is to form 6-carbon citrate is enzyme related. So it's talking about enzymes here more than anything, and it's asking mainly how do you know how does it enable acetyl coenzyme A to then bind to the enzyme. Well, in terms of the enzyme, it's the shape of the active site is being changed. So the active site is moulding around the substrate, okay? So therefore it's now complementary. Alright, so you could also say that there's a change in the tertiary structure affecting the active site. Key thing here is to look for the clues. This looks a very complicated question, but when you look at the clues, for example, it talks about enzyme catalyzed by enzyme reactions, okay? So it's very much, you know, if you look at that answer, you will know the answer, but you've got to look at the clues in the question. Case 6bii, it talks about succinyl coenzyme A, and I'm pretty sure we haven't really discussed that. But it does give you a clue. It says it has a very similar shape to acetyl coenzyme A. And it talks about how this succinyl coenzyme A could control the rate of the reaction. And really it's looking at the fact that it's a competitive inhibitor. Okay, so I know that's kind of synoptic in certain elements, but it's a competitive inhibitor which then prevents the enzyme substrate complex forming. So be aware that they may talk about other aspects from your learning in different topics. Six C is checking on your understanding of anaerobic respiration. So 6CI is looking at why does converting pyruvate to lactate allow the continued production of ATP during anaerobic respiration. And basically, you should know, if you, again, this is kind of learning basically in your books and your notes, it regenerates. It basically oxidizes reduced NAD, and that's used in glycolysis. 6C2 is looking at the fact that some of the lactate is converted back to pyruvate and when they're supplied with oxygen. So one advantage, basically it can enter the link reaction, or it's used, I think the main answer is it's used in aerobic respiration. Also, you could have said that it reduces cramp or fatigue as well. Notice that you've got to talk specifically. Don't say things like it reduces muscle ache. You've got to be very specific that it reduces muscle fatigue. Okay, now on to a big question here. Question 7, 15 marks. And this is really important to say that your grade boundaries are so small that this type of question can change your grade dramatically. It's really important that you look at all the evidence, all the information that they give you. You will get a 15-mark question. You'll get two 15-mark questions. This one is very much about evaluation, summarising conclusions, thinking about the information that they give you. So it's really, really important you practice this style of question if you found it difficult this time around. So question 7a is asking you to suggest how climate change could cause an increase in soil salinity. So it's asking for why would soil salinity increase, as it has mentioned in the stem of the question. The first obvious one is an increase in temperature, okay? But you then need to go on to get the second mark. You need to say, well, how will that affect soil salinity? And well, basically, you'll get evaporation of water from the soil. So if you get evaporation of water, you get higher salinity. It's as simple as that. Question 7b, it asks, explain why crop plants do not grow when soil salinity is 2% or higher. Now, this type of question is, on the face value, if you're in an exam, I can understand why this might confuse you. But you've just said previously about the fact that with soil salinity there's an evaporation of water. So you need to think, well, if there's less water in the soil, then if it's to do with water, my first kind of thought is water, movement of water has to be something to do with osmosis. So the answer is basically asking you to mention that basically soil has a reduced water potential, so you need to mention water potential, okay, and the plant itself has a higher water potential, therefore through osmosis you'll get movement of water, diffusion of water from the plant into the soil, okay. So this answer here, this is sufficient to get the 2 marks. Again, I did notice many of you were writing many more lines of text to try and explain. You could also say that plants or crops take up less water, which is fine, but that's the general answer, and you need to kind of think a bit outside the box when you're answering that style of question. Okay, so 7c. Now here is, in the exam, this is where you need to spend a tiny bit of time looking at the information. They give you a lot of information about cucumbers that are grown in soil with salinity at 1.5%, and then they talk about a fungal disease, CFRR, and they talk about 4 species of bacterium being used as a biological agent. So there's lots and lots of things going on in this question. You need to, again, I wouldn't get too concerned about the fact that you don't know what the biological agents are. It's more about the information that they give. They then talk a bit about the methodology of how they got the results. Okay, and they talk about the treatments. Cucumber seeds were coated in one of the 4 species of bacterium. And they talk about control as well. So there's lots and lots of information there. They also have some statistics as well. So expect stats within your paper, okay? So here they've got standard error, okay, between them. But you need to spend a tiny bit of time working out what's going on in the data. Okay, so first question 7CI. Explain why the CFRR fungus was isolated from several cucumber plants and why these plants were grown in salinated soil. Well, what they were looking for here was the key thing there, as I said, is several cucumber plants. If you collect it from several cucumber plants, then basically you're going to obtain different strains from different types of cucumbers. Okay, so it's ensuring you collect a variation of CFRR. In terms of growing in salinated soil, well, that's the natural normal conditions they're normally grown in. That is in the stem of the question. Now in this question, unfortunately, some of you did kind of get the wrong end of the stick about when fungus was isolated from several cucumber plants, because some of you thought that it was a method of preventing the spread of the disease, or another misconception was that it suggested that collecting from several plants ensured that the fungus was the same, which it is not. It's from different, you get different strains of fungus from different types of plants. Okay, so 7C2. This is a really effective discriminator for your examiners. Okay, you will be expected to evaluate the effectiveness of something or other in the exam. In this case, it's the effectiveness of bacteria as biological control agents. The better answers kind of split that up, rather than kind of going straight in and giving lots and lots of information that seemed a bit higgledy-piggledy. People who got the full marks were really clear with their points, and I think it's really important when you write your answers, you've got to remember your examiner, i.e. a teacher, i.e. someone like myself or Mrs. Miller, for example. When we're looking at the answers, we're going to, if we can see a well-structured answer, it's just psychologically better in terms of giving full marks and giving benefit of the doubt. So make sure your answers to the longer answer questions are really succinct, to the point, and they're organised. If you look at the mark scheme, the first key thing is looking at the data as well, is that the number of disease plants is decreased by all bacteria. That's the first main point. You would then need to go on and actually talk about which were the most effective bacteria, which were the least effective. So in this case, again, if you look at the mark scheme, you don't need to put Serratica rhizophylla. You put S. rhizophylla is most effective and S. plymoutheca is least effective. So you want, that's an evaluation, isn't it? It evaluates rather than just saying one or the other. The next big thing is talking about the standard error. They wouldn't be showing standard error bars unless they wanted you to discuss them. So in terms of standard error, it mentions nothing about what is standard error. So I would mention it in your evaluation that if basically there's no overlap of the standard error, then it indicates a difference is significant. That's all you need to do. Or you could say the opposite, that if standard error does overlap, then indicates there's no significance. And then you need to give an example. Obviously it's only 4 marks, you don't have to describe every single minutiae of detail, but you need to mention maybe something that is significant and something that isn't significant. So for example, you could talk about some of the bacteria, so overlap of standard area of S. plymoutheca with the control, or for example, there's no overlap of the standard error for S. rhizophylla with the control B. So, you know, if you break that down, it's not a difficult question, but it is, for the examiners, that is a really good discriminator. And it's important you take your time to answer those kind of things correctly, because it's worth 4 marks, and it's just about evaluating the data that they give you. Okay, question 7C3. Before we go on, you really do need to know why you have a control in an experiment. Okay, and this is a really good example of this style of question. The first thing, most of you got the 1 mark at least, knowing that it's a comparison. You're comparing, so you need to make a comparison between control A and the treatments. The second mark, though, a lot of students didn't then go on to say, well, it's basically then to see the effect of adding fungus or having no fungus. It shows the effect of fungus in the soil. That will get you the 2nd mark. Okay, question 7D is kind of taking a different direction now with this question, and you find this with the 15-mark questions. They don't tend to stay on the same level, and then they're going to be just testing something else. In this case, it's talking about bacteria and how it may stimulate an increase in dry mass of cucumber plants. So you need to think, well, where else would I find bacteria in terms of helping plant growth? And you need to kind of use your Philodex of information in your brain and think, well, it's something to do with the nitrogen cycle. So you know that bacteria are nitrogen fixing, okay? So you need to think, well, they're probably nitrogen fixing, and therefore that would increase the mass. You could also think about it reduces infection possibly, or it produces growth stimulating substances. So again, you don't have to talk about nitrogen cycle, we can talk about other things. But it does say suggest. Most of the incorrect answers to this question were related to the fact that they were saying that bacteria is producing extra carbon dioxide from basically increased photosynthesis. Again, that's obviously incorrect. Final question, 7D2, it's talking about growing cucumbers plants in pots. They're saying that they remove the weeds. Why did they remove these weeds? Well, we know weeds are going to compete, well, plants are going to compete for different things, so you can say that they compete for minerals, salts, ions, nutrients or light. But then again, remember, it's 2 marks, so what effect would that have? Again, lots of people miss this 2nd mark, and it's just simply saying that it would reduce the dry mass of the cucumber, because obviously they're taking the nutrients away from the cucumber. So that's your 15-mark question there, and I think, as you can see, it's a really good example of the fact that you've got to use different skills, evaluation, summarising information, using your knowledge in unfamiliar ways. Okay, so you need to think carefully of what kind of answers. They can ask you pretty much anything, and as you can see, you even had a bit of statistics in there as well. So what I suggest you do, if you found that question difficult, please use the exam bank that is in the 6th Form Centre, and you can practice all the Unit 4 15-mark questions. Okay, on to question 8, and again another 15-mark question, this time split into 3 longer answer questions. So this is a really good opportunity to show all the revision you have done. A lot of these questions are basically learn and churn, and so they obviously want specific detail, but these are the kind of things that you can really revise. These longer answer questions, you shouldn't be a problem if you have done effective revision. So question A is all about isolation of species and how new species could have evolved, and it's really clear that, again, some of you missed some very easy marks, and I would put that down to a lack of revision or not using key terminology at times. So all that's looking for is on key terms related to, for example, many of you forgot to mention geographical isolation. Okay, so not using that word meant that you lost one of those potential 6 marks. Most of you went on to looking at variation due to mutation. You looked at the fact that the gene pools were separated, there's no interbreeding, different selection pressures because of different conditions or habitats. Then reproductive success is different, and then it leads to a change or an increase in allele frequency. If you look at the mark scheme as well, they will, I mean, there are different marks you can get. Obviously, naming a abiotic or biotic condition, or saying that alleles are passed on to the next generation. But I think you also need to, I think many of you weren't saying that basically you'll get an increase in allele frequency. Question 8b, it was basically looking at your knowledge of mark release recapture method to estimate the numbers of lizards. On the whole, this was very well answered by students. Most of you realising that you have to capture, sample them, mark and release. Many of you realising that the method of marking, it was important that they weren't visible to predators. I think the third mark that many of you didn't get was saying that you've got to leave a sufficient time for the lizards to be distributed, randomly distributed or distributed evenly. So you didn't specify the fact that you would leave it for a sufficient time and therefore lost that mark. And then many of you use the equation, which is brilliant because the equation is not in the stem of the question, and so that can be used. Okay, there's the answer. There's no additional commentary related to that, and it's quite a nice simple answer. Okay, 8C, again, this in the examiner's report they say that this was a really good discriminator, providing a good spread of marks. I think there were some really good answers here, providing detailed accounts of the relative effects of photosynthesis and respiration on the concentration of carbon dioxide over 24 hours and at different heights. However, I think some of you kind of, again, because you didn't talk specifically about what they wanted, which was over the 2 bullet points, and I think some of you missed certain marks there. So the kind of answers you were looking at was basically with a higher concentration, increase in carbon dioxide linked to respiration in the night. There's no photosynthesis in the dark because it's over the periods of 24 hours. In light, net uptake of carbon dioxide is, they use more carbon dioxide. And there's I think at different heights you need to talk about different concentration of carbon dioxide as well. And there's fifth one, at ground level, less photosynthesis, there's less light, there's more microbes as well producing carbon dioxide. More difficult question that, but a good discriminator.